Hello students, today we will be studying Histology of Lymphoid Tissue Part 2. In Part 1, we have seen the basic information about the lymphoid tissue and the various cells that are seen in the lymphoid tissue. In this video, we will be seeing the two important slides of lymphoid tissue, that is the lymph node and the thymus. Lymph node. It is a small kidney-shaped structure that filters lymph. It shows a hilum on its concave surface. It receives lymph through many afferent lymphatics, and after filtering the lymph, it sends the lymph through efferent lymphatic via the hilum. Blood vessels enter the lymph node through the hilum. Histologically, the lymph node shows capsule and parenchyma. Capsule is the outer covering of the lymph node made up of dense connective tissue. Trabeculae extend from the capsule into the substance of the lymph node. Parenchyma of lymph node shows outer cortex and inner medulla. The cortex shows darkly stained superficial nodular cortex having numerous lymphatic follicles containing B lymphocytes. These nodules have a central lightly stained area called as the germinal center, and this is the zone of the B cell proliferation. And the cortex shows deep cortex or paracortex that contains T lymphocytes. Hence it is called as the thymus dependent cortex. The medulla of the lymph node shows presence of medullary cords, which are anastomosing cords of lymphocytes, macrophages and plasma cells. And it also shows medullary sinuses, which are spaces between adjacent medullary cords that converge at the hilum to form the efferent vessels. The medullary cords contain B lymphocytes, plasma cells and macrophages. Reticular meshwork, that is the supporting framework of the lymph node, consists of reticular cells, reticular fibers, follicular dendritic cells and macrophages. The reticular cells synthesize type 3 collagen fibers and reticular fibers. Dendritic cells present the antigens to the T lymphocytes. Let us now see the sinuses of the lymph node. Three types of sinuses are seen here. Subcapsular sinus lies just beneath the capsule and it receives lymph from the afferent lymph vessels. Trabecular or intermediate sinuses, these are present adjacent to the trabeculae and they connect the subcapsular sinus to the medullary sinuses. Medullary sinuses lie among the anastomosing medullary cords. They receive lymph from trabecular sinuses and drain it into the efferent lymph vessels. The sinuses are filled with reticular meshwork that helps in filtration of lymph, trapping of cellular debris and microorganisms, and antigen presentation to immune cells. Sinuses are lined by endothelium and reticular cells. Let us now see the circulation through the lymph node. The afferent lymphatics bring the lymph and pour it into the subcapsular sinus. From there, the lymph goes into the trabecular sinuses, then into the medullary sinuses, and then all these medullary sinuses converge towards the hilum, and that is how they drain into the efferent lymphatics. What is the function of lymph node? Filtration of lymph, presentation of antigen to T lymphocytes, activation and proliferation of B lymphocytes. Also, conversion of B lymphocytes to plasma cells and production of antibodies, removal of bacteria, support of humoral and cell-mediated immunity. This we see a low power view of a slide of the lymph node, and what we can see here is the outer capsule. Deep to it we see the parenchyma showing cortex, paracorticular zone and medulla. Cortex, as we can see here, shows lymphoid follicles having a pale central germinal center. A slightly magnified view of the same shows us the capsule made up of dense connective tissue, the subcapsular sinus which lies just beneath the capsule, and the outer area of the cortex which shows the lymphoid follicles, and these lymphoid follicles have a central paler area which is the germinal center, and this germinal center is where proliferation of the lymphocytes takes place. This what we see is the medulla of the lymph node, which shows cord-like aggregations of the lymphocytes, and those are the medullary cords, and in between them we see the medullary sinuses. The salient features for spotting for a slide of lymph node are presence of subcapsular sinus, presence of cortex showing lymphatic nodules, presence of medulla rich in blood vessels. Next we see the slide of thymus. Thymus is a primary lymphoid organ. It is a bilobed gland situated in the anterior part of the superior mediastinum. It has capsule, cortex and medulla. The capsule is a thin connective tissue capsule that covers the entire gland. Trabeculae and septae extend into the parenchyma and divide the gland into incomplete thymic lobules. Each lobule has outer cortex and inner medulla. The septae are shorter and they do not traverse the entire parenchyma, and hence medulla of adjacent thymic lobules is continuous with each other. The cortex of a thymus lobule is the peripheral dark basophilic zone of the lobule containing closely packed T lymphocytes known as thymocytes, supported by a meshwork of epithelio-reticular cells. Cortex also contains few macrophages that phagocytose the degenerating lymphocytes. The medulla of the thymic lobule is the inner lighter zone having lesser number of thymocytes, and it also contains epithelio-reticulocytes and the Hassel's corpuscles. The Hassel's corpuscles or thymic corpuscles are small, rounded or oval structures present in the medulla of the thymic lobules. They are made up of eosinophilic hyaline mass at the center that is surrounded by concentrically arranged type 4 epithelio-reticular cells. The Hassel's corpuscles represent degenerating thymocytes, and their number increases with age. Thymic corpuscles secrete thymosin and thymopoietin hormones. What are the functions of the thymus gland? Maturation of T lymphocytes. They provide an isolated environment for T lymphocyte maturation. Secretion of thymopoietin by epithelio-reticular cells, and this thymopoietin stimulates the T lymphocyte production. Thymus is essential for development of immunity in the early life. After puberty, the thymus undergoes gradual atrophy, and that is called as the thymic involution. Blood thymic barrier. Immature T lymphocytes mature in the thymus in an isolated environment. Blood thymic barrier prevents the exposure of T lymphocytes to the antigens. This blood thymic barrier is formed by capillary endothelium with basal lamina and few pericytes, perivascular connective tissue with few macrophages, and type 1 epithelio-reticular cells. The blood thymic barrier isolates the T lymphocytes from circulating blood and antigens in it. This here we see a low power view of the slide of thymus. We can see a capsule made up of thin connective tissue sending in trabeculae or septae, which are short and incomplete septae, and that divides the thymus into a number of thymic lobules. Also seen here is how the septae are incomplete and thus the medulla of adjacent thymic lobules can be continuous with each other. A magnified view of the same shows us the individual thymic lobules having outer darkly basophilic stained cortex and a central lighter medulla. What we also see here is the medulla showing presence of the Hassel's corpuscles, which are seen as eosinophilic material. Basically, these are the degenerating epithelio-reticular cells. The salient features for spotting for a slide of thymus are presence of outermost capsule with incomplete septae, presence of thymic lobules with cortex and medulla, presence of Hassel's corpuscles in the medulla. Thus, we have seen the two important slides of lymphoid tissue today, that is the slide of lymph node and thymus. In the next part, we will be seeing the other two important slides, that is spleen and palatine tonsil, and we will also then see how to differentiate between all the four slides of lymphoid tissue. For further reading, you can go through Sontakke Yogesh, Textbook of Human Histology by CBS Publishers and Distributors Private Limited, New Delhi, 2020, page numbers 106 to 121. Thank you.